Hi, my name is Hedges. I'm a PhD student at the University of Adelaide in South Australia. During my PhD, I'm looking at the conservation and management of Australia's granite rock holes. I'd like to begin today by acknowledging the Kukatha, Wirangu and Barngarla people, who are the traditional custodians of the land on which I undertook the field component of my research. I'd like to also acknowledge the Kaurna people of the Adelaide Plains, on whose land the University of Adelaide now lies. I'd like to pay my respects to their elders past and present, and I extend that respect to Aboriginal and Torres Strait Islander peoples here today. Now, the rock holes I'm researching are present wherever flat granite explodes above the surface of the soil, so can be found across much of southern Australia. They fill up following rainfall, and in doing so a unique ecosystem forms. Invertebrates hatch out of eggs that have been lying in wait in the sediments. This is after having survived months of being baked in the Australian summer heat. Aquatic plants grow from seeds that have also adapted to these harsh conditions. But why are these rock holes important, and why am I interested in them? Well, first and foremost, they're an important source of fresh water to a number of species of conservation interest. Animals move long distances across the otherwise very dry Australian landscape to find water, and so if such sites aren't maintained, their degradation may pose a serious threat to proactive conservation attempts. There is also a lot of anecdotal evidence that rock holes are being used by a number of invasive species. So understanding when and how they're being used by animals like goats, foxes, and feral cats may help us manage these species more effectively and decide where our efforts are best placed. They're potentially an important part of the life cycle of some of Australia's less understood species, like the desert burrowing frog, meaning that we can use them to observe these animals and better understand their biology. Being rain-fed, they're also weather-dependent, so they're potentially vulnerable to the impacts of climate change, particularly in areas that are projected to experience a significantly drying climate over the next 100 years, like much of southern Australia. My primary research sites are the rock holes at Hedges Nature Reserve in South Australia. Though there are many other sites across Australia with similar rock holes, some of which I've visited over the last few years. I had hoped to get to more rock holes during my PhD, but state border closures and travel restrictions have made this difficult. Hedges Nature Reserve is managed by South Australia's Nature Foundation and is home to a number of species of conservation importance, such as the yellow-footed rock wallaby. The Nature Foundation has been fantastic in allowing me access to this land, facilitating my research, offering guidance and support, and providing generous research funding to my PhD. Granite rock holes, sometimes referred to as namas, a Nyungar word from Western Australia, have long been used by Australia's First Nations peoples as a reliable source of fresh water. In the past, their locations were recorded in carvings and through songlines. The rock holes at Hedges are still managed today, using traditional techniques by the people of the Gawler Ranges Aboriginal Corporation and their Kids on Country program. You can see the use of timber and stones to prevent animals from falling into the rock holes and fouling the water, as well as limiting loss to evaporation. The rock holes vary in their size and shape, distance from each other, and the length of the period for which they stay wet. Here's an example of the size of one of the rock holes. You can see me lying down next to it. Over the last 2 years, I've undertaken 6 research trips out to Hedges, supported by a team of volunteers. Some of these trips have had to happen at the drop of a hat after an unexpected rainfall event, so I want to thank all 13 of my field assistants for being so dedicated. During my first trip, we located over 90 individual rock holes, recorded their location, and painstakingly measured their morphology using this awesome state-of-the-art measuring pole I designed. This took about 6 days of 9 to 5 work on top of swelteringly hot rocks. Then I won a grant awarded by the Lake Eyre Basin Authority and decided to throw out those measurements in favour of some much cooler measurements. I brought Dylan Campbell from Adelaide Uni's Unmanned Research Aircraft Facility out to Hedges with me, and he flew his actually state-of-the-art imaging drone. He flew overlapping transects over the rock holes, imaging each rock hole from hundreds of different angles using standard RGB sensors mounted on the drone. Using photogrammetry, we produce 3D models of each rock hole, allowing us to more accurately measure the rock hole size, shape, catchment area, and more. I'm using these models elsewhere in my PhD to do hydrological and climate modelling to assess the vulnerability of these ecosystems to the impacts of climate change. We then place motion-triggered wildlife camera traps in the field, pointed at a series of rock holes. The idea here is that every time an animal approaches a rock hole, the cameras will trigger, a photograph will be taken, and this will record the visitation event. We're hoping we'll be able to work out what vertebrates frequently visit the rock holes, and that we'll be able to test whether different animals have preferences for the sort of rock hole they visit. I had these camera traps deployed for almost a year in total, though not all of them survived the harsh desert conditions for this entire period without failing. Temperatures out there get really hot, then really cold, then really wet, then really dry. Moments after deploying this particular camera, Joe and I got caught in a hailstorm and had to shelter in granite caves until it passed. The cameras recorded visitation events by a large number of different terrestrial Australian vertebrates. Echidnas, wombats, kangaroos and emus were all commonly recorded visiting the rock hole. A lot of flighted bird species were also recorded, though it's a little harder to confidently confirm each of these photographs as a true rock hole visitation. We also caught a bunch of pest species accessing the water too. Foxes, invasive goats and rabbits were all common, with a few feral cats showing up too. I've been using 2 different techniques to process these images. The first is a machine learning algorithm called megadetector, which processes the photo in bulk, sorts out false triggers, and groups similar looking animals together. The second is, well, undergraduate students. A huge thanks to Ashton, Brianna, Flynn and Peter for processing literally thousands of photographs as part of their Conservation Biology 3 research project. So our first preliminary finding, and I stress preliminary because I haven't done the full morphometrics yet, is that rock hole morphology doesn't predict vertebrate attendance at all, really. So my first preliminary recommendation is that you just throw away your state-of-the-art photogrammetry capable drone. Our next finding is a little more interesting. There seems to be some pretty clear partitioning of rock hole use by native vertebrates based on altitude, or at least the slope an animal will need to have travelled across to reach the rock hole location. The low-lying rock holes were used by wombats, emus, and 3 different kangaroo species, whereas the rock holes at higher altitudes were used by wedge-tailed eagles, echidnas, and only 1 species of kangaroo. Unsurprisingly, most of the parrots and other smaller birds were recorded at rock holes of any altitude. Use partitioning was also observed to a degree when considering invasive vertebrate species, but the pests of highest concern at Hilltipper, feral cats and goats, don't seem to be concerned with altitude, with goats being recorded as having visited almost every rock hole photographed, and often in really high numbers. So we know now that the rock holes are being used by vertebrates at Hilltipper, and that there is some discrimination between sites, but there are some serious limitations to the data we can generate with camera traps. Firstly, you end up with a lot of false triggers, which slow down your capacity to process data. Machine learning and an undergrad army can help with this, but they can't handle everything. IDs aren't always easy. Often photos, particularly nighttime photos, look like this. Pictures like this are really hard for even experts to ID, especially in locations that have similar looking species, like Hilltipper, which has 3 different species of kangaroo plus wallabies. This particular photo led to one of my undergraduate students sending me an email with this exciting subject line. So maybe this photo is my ticket to an easy nature paper. I also had difficulty placing camera traps. There are no trees on the outcrops that you can attach them to, and you can't exactly hammer star droppers into granite, especially with their cultural and geological significance. I was limited to placing the camera traps around the outside of outcrops, which meant the photos weren't always ideal for accurate IDs. It's also a long 8-hour drive each way to the nature reserve, and I don't want to be driving all the time to check camera traps. It's expensive, I have to use a lot of diesel, and I'm struggling to find new podcasts to listen to. So I started thinking, what if there was some technique that could be used by the volunteer rotational managers on site to detect when vertebrates were visiting rock holes and other water points for fresh water? We're currently in the early stages of developing an environmental DNA tool for detecting vertebrate visitation to rock holes. The idea behind this is that every time a vertebrate visits a rock hole, they leave behind a little trace of DNA that sits there in the water. This eDNA can then be collected I've been collecting water samples during my PhD and ran 3 collecting trips last year where I did this. From each rock hole, I took 5 1-litre water samples plus a field blank to be able to test for any contamination. I also made sure to always pose in unflattering positions whilst collecting my samples. I then filtered these samples through membranes with a pore size of 0.2 microns. This allows the DNA to be retained and the wastewater to be discarded. I've used 2 different systems to do this, both of which involve applying a vacuum to the sample to draw it through the membrane. Then we freeze the membranes and bring them back to the lab for further processing. The pipeline we follow then looks a little like this. You take your frozen membrane and you run a standard extraction across it. You then quantify the DNA using qPCR and apply a series of assays that amplify DNA within the 18S region for vertebrates. We're then undertaking eDNA sequencing, and we're doing this in collaboration with eDNA Frontiers at Curtin University. This allows you to generate your DNA sequences, which should correlate with all of those little fragments of DNA that are present in the sample. You then blast this against the database of sequences of known origin. This allows you to generate species IDs for all the animals that have visited or lived in that water sample. Now, this is where I had hoped to be by the time of this talk, and when I submitted my abstract all the way back in March, I confidently thought, yes, I'll be here by now. But instead of all of us being able to party together in New Zealand, we're presenting over Zoom. The pandemic has similarly impacted my research, so unfortunately, I'm still stuck at this stage, waiting for DNA sequences to come through. Now, I have still gotten quite a bit done over the last 2 years, and I'm confident that this study has brought me closer to answering the overarching question of my PhD, this being, what are the mitigatable threats to the role of ephemeral freshwater bodies in the Australian environment? I'm adding to our understanding of ephemeral freshwater body ecology, particularly in Australia's arid and semi-arid regions. I'm producing a species visitation list, detailing species that I've observed visiting the freshwater rock holes at Hedges. I'll be providing conservation recommendations to Nature Foundation, which will likely focus on the need for further suppression of goat populations, though if this is not matched at a landscape level, it's not likely to be very effective. I've also helped to improve our knowledge of the specific value of granite rock holes to local vertebrate species at Hedges Nature Reserve. So I'd like to thank everyone that's contributed to my project in some way throughout my PhD so far, my various funding bodies, the Nature Foundation, the Australian Government, the Ecological Society of Australia and the Environment Institute, my supervisors Phil, Andy and Michelle, and all of my collaborators, my lab group and everyone else who supported me over the past 2 years. Thanks. Hopefully I'll have a few minutes now to answer some questions. Otherwise you can contact me through the details on the screen there now. Thank you.